data. Is that right? No, it's this one. Lab data entry. So if you click there, it's smaller. These are all of the labs for which you will be inputting your own data so that I can compile all the class data and then I will send them out to you. So notice it says bacterial ubiquity. If you click on that, you will come to a Google form where you'll put your student number, right? That's the number that's on your desk. And then you will put your plate exposure method. So all of those different methods are there in the drop-down menu. And, right, so that was a while ago we collected those data, but remember I said hang on to them because we're going to keep them together. Then how many colonies you saw. And then broth, right, half of you rubbed a body part, half of you rubbed some inanimate object, so you choose one of those. And then you'll tell us the specific site. So my cell phone, the doorknob, my earlobe, wherever it was that you did that. And then you can check any or all of these, right? So you should at least check one thing. Even if it didn't work, like I can't remember who it was, but somebody had a big furry mold growing on the top of their broth. And was it? Okay, I knew it was over here somewhere. We won't ask her where she swapped it, right? But we said we're not going to open that because remember I told you we don't open molds because molds, the spores can go into the air. So you would just put, right, inconclusive. So for some reason you couldn't do the gram stain, you would say inconclusive. If you did the gram stain and there were no organisms, there was another person that had that, right? They had very little turbidity in the broth and we couldn't get anything on the gram stain. So everyone will check at least one box. And then those of you who got results, if you got gram-positive cocci, you will check that box, gram-positive bacilli, gram-negative cocci, gram-negative bacilli, spore formers. We have lots of spores. And I know you're thinking, well, spores, we haven't done spore staining, right? But because a spore is really highly resistant to penetration, the gram stain reagents, the stains can't get into it. So we had a lot of people that had purple cells with a clear oval inside them. So you will choose those things, click submit. When you click submit, you'll know that your data were submitted. Well, this is already, but it'll pop up that says your data, been sent off. So you will know that I got your data. So your data are due by 6 o'clock tonight. You should have time to do that in class. So if your data are not in by 6 o'clock tonight, then that could be a place where you would lose a lab technique point, right? So make sure your data are in by 6. When everyone's data are in, then I will send out a sheet for you guys that you can print and put into your lab manual so that you have all of the data together. And Amy.
The lab manual says that if you have over 100, just put like 100 plus. So should we just put 100?
Yeah, you can put 100 plus. That's fine. Yeah, it'll allow you to put something that's not technically a number, I believe.
I'll make sure.
If not, I can fix it. I'll check it before when we take a break today. I will check it and make sure. But yeah, you can put 100. Or if you want, if it won't, you can just put like 1,000, and we'll know that that meant that you weren't able to count them. Because one thing that you'll be learning soon in class is that we never count more than 300 colonies on a plate. So whenever you see more than 300, that's just kind of there were so many, we don't know how many there were. All right, and Shadi, you had your...
So I did this before you explained it. I thought it was due beforehand.
Okay.
But now that you've explained it, I realized I made a mistake. Is there any way I can go back?
Oh, just re-upload it. So if that ever happens to you, if you accidentally hit submit or something happens, I can see a timestamp of when you upload your data, and if I see 2 of them, I will delete the earlier and assume that the second one you sent me is the one that was right. So if that ever happens, because as you can see, right, there's going to be quite a few days where we're uploading data. If you ever make a mistake, just do it again, and I will see the earlier and delete it and keep the later data. Okay, well, I hope you all enjoyed your day off yesterday. Because of the fact that we have a Monday holiday, this might have happened to you in the past with lab classes. When we have a lab that runs for the whole week, if there's a Monday holiday, Tuesdays will be kind of weird because we don't want to come off schedule and have people in different classes doing things on different days. So we don't have a lab for today. So what we do is use that as time to just get more lecture in, because that allows me to give you a bigger buffer between when we're done with lecture material and we have the test. So you get more time to study that material rather than, you know, a 3-day turnaround between test or end of material and test, you get a week. So we're going to finish up lab 5 today, or chapter 5 today, take a break, and then we'll do chapter 6. And then what I thought we'd do, just so we can kind of have a clean slate on Thursday, is just get your motility media and your swarm plates out so that then we can be done with lab 7. And you don't have to have another thing that you're holding on to that we're partially finished with, right? Because you did your wet mount, and now you'll be able to get your other 2. So it won't really be a lab, no lab quiz or anything like that, but you'll just collect those data and then you can go home. So, all right, I think that's, yes, that was my little note for myself. Okay, so we talked all about ATP generation, right? So we have substrate level, we have that electron transport chain, right, which is oxidative phosphorylation, all of that good stuff, and then we switched and talked about heterotrophy, right, which is the eaters. Now today we're going to talk about autotrophy, which is the self-feeders, right? Rather than having something else feed you, you feed yourself by making your own food. But just to recap, right, those questions, right, that we ponder when we're talking about heterotrophy, right, we ask a couple of questions, and all of those organisms have these answers in common. That's why we call them heterotrophs, right? And the big one is, what kind of carbon do you use? Do you use inorganic carbon, in other words CO2, or do you use organic carbon? And all heterotrophs, right? I mean, you might be a vegan, a vegetarian, a pescatarian, a carnivore, like you might be all sorts of things, but you all eat. And so you're all consuming organic carbon as your carbon source, right? So that's your food. Where do you get your electrons? Also from your food, right? This is your food. It gives you your carbon, and it gives you all of your electrons, right? So for us, for heterotrophs, the carbon source, the electron source are the food that we eat. What do you do with the electrons, right? Ultimately, we take all of these electrons, right? We oxidize that glucose molecule completely to CO2, and we pile those electrons onto oxygen, and we throw those electrons away because we're done with them, right? And Nassim was our oxygen molecule, right? She collected the electron, and then she went away, right? That's why you have to breathe, because we need a new oxygen molecule to land into that spot. Right? And then we learned that for E. coli, that's not always the case, right? E. coli can get rid of oxygen and bring in nitrate or carbonate or sulfate or some other molecule to serve as an electron acceptor. So we learned that this is not the only kind of respiration, right? This is what we would call aerobic respiration. And the other kinds of respiration where some other inorganic molecule sits here are anaerobic, right? Not oxygen requiring. So we talked about all of that. And then we said we were going to talk about autotrophy, right? And in general biology, autotrophy is synonymous with photosynthesis. When we think autotrophy, we think plants doing their beautiful thing where they make food for us and we get to eat that food, right? So they use sunlight, and they do this magic with it, right, in their chloroplast, where they convert sunlight to the chemical energy of ATP and use the chemical energy of ATP to fix carbon or take CO2 and build it into food. Now, what I would like to point out for you, well, first of all, is this isn't the only kind of photosynthesis. So we're going to build on photosynthesis in this class. But not only is there a requirement for energy for this making of food to occur, you also have a requirement for electrons. Because really when we make food, right, when we take CO2 and fix it, right, we can call this fixing carbon, right? Fixing carbon means taking inorganic carbon and converting it to an organic molecule, but what we also could call that is reducing carbon, right? Because this carbon has no electrons for sale on it, and this one has a ton. So we could also call this reduction. And we know, and this is why we made such a big deal about talking about electrons moving in molecules, we know that reduction means the movement of electrons, right? Which in biological systems, because we're messy, means H's. And you need H's to make that happen. So you need ATP and you need electrons, right? And electrons, we watch them in the form of H's, right? Because that H is that special little atom that has just a proton and an electron. So this is photosynthesis, right? Now, we more particularly call this photoautotrophy. Autotrophy, feeding yourself, making your own food. What's fueling you making your own food? The sun. Why are we making that a distinction? Because it turns out we also have chemoautotrophs, organisms who make their own food, but they don't use sunlight to do that. They use chemical energy to do that. So like you, they're a chemotroph. They eat chemicals, but they eat inorganic chemicals, and you eat organic chemicals. You're a chemoheterotroph, and we're going to look today at chemoautotrophs. So when we talk about autotrophs, there is some common ground, because the autotrophs are all making food, right? So if you're making food, you need two things. You need ATP and you need electrons. So regardless of whether you're a photoautotroph or a chemoautotroph, we can ask some general questions that all of you can answer, right? So we can say, what kind of carbon do you use? And you would say, I eat inorganic carbon, CO2. Where do you get your electrons? It's always an inorganic molecule, right? What do you do with your electrons, right? This is different. Heterotrophs throw their electrons away. Autotrophs save them. So if we look at those, right? So energy in some form, right? Chemical energy, light energy. Here's your carbon source. Here's your electron source. These are both inorganic molecules. CO2 is your carbon source. Water, in this case, is your electron source, right? Meaning we take electrons from water and we pile those electrons onto CO2. So we oxidize water to reduce CO2, and we're left with, remember that was our abbreviation for sugar, and water and oxygen. And we learned back last week that that's not just called photosynthesis, that's called, right, oxygenic photosynthesis, because it generates oxygen here. And what we're going to learn today is that there is also anoxygenic photosynthesis, not oxygen generating photosynthesis. So the goal today is to understand autotrophy, and we're going to do that. It's going to be kind of funny. We're going to do that by starting at the end, because the end is what everybody has in common, right? What they all have in common is this right here, this arrow. So we're going to start with the end. We're not going to start with how do you make energy? Where do you get the electrons? We're going to go right to here, right? So we're going to skip 1 and skip 2, and we're going to answer the question first. What do you do with your electrons? You make food with them. What does that look like? What does that mean? So we're going to start at the end, because that's what everybody who's an autotroph has in common. This right here, inorganic carbon to organic carbon, because that technically is what it means to be an autotroph. So if you are an autotroph, you are a CO2 reducer, or you could say you're a CO2 fixer. Those are all terms that mean exactly the same thing. You take inorganic carbon in, and you give off or release organic carbon. Now, by far, the very most common way that any organism on the planet takes in CO2 and converts it to a sugar is something called the Calvin-Benson cycle, right? Oftentimes also called the Calvin cycle. Plants, protists, all eukaryotes who are capable of autotrophy undergo the Calvin cycle, right? Named for two people, Calvin and Benson, right? Also called the, right, tricarboxylic acid cycle, I think, or something like that, right? All of those things mean exactly the same thing. They mean that you take in CO2 and you release fructose, which is a sugar. And if you don't believe me, you can look down here, right? There's lots of crazy stoichiometry here, which is most people's least favorite part of chemistry. But we don't care about all the numbers, but CO2 to a C6H12O6. That's got lots of H's. It didn't used to have H's, now it has H's. It's been reduced. That's the food that results from the Calvin cycle, from fixing CO2. So all plants, all protists, all eukaryotes, and quite a few bacteria use this right here in order to fix carbon. Scratch what I said about TCA, just Calvin cycle. Sorry, I got ahead of myself. The other two you've probably never heard of because notice they're only done in bacteria. And that's not our focus in general biology, right? General biology, we got to focus on bigger things. So you only hear about the Calvin-Benson cycle because that's what the plants do. But in microbiology, we are going to talk about these two as well, right? So we have two additional pathways for fixing carbon. They are the reverse citric acid cycle and the hydroxypropionate pathway. Now, if you're wondering why the reverse citric acid cycle, the reason this is the reverse citric acid cycle is if you knew a lot about the Krebs cycle, you would notice this is the exact reverse of the Krebs cycle. You think, Krebs cycle, what does that have to do with photosynthesis? Krebs cycle has nothing to do with photosynthesis. Krebs cycle has to do with taking an organic molecule and oxidizing it completely to CO2. But what's the reverse of that? Taking CO2 and reducing it to an organic molecule, right? So they call this the reverse citric acid cycle because the Krebs cycle has an alternate name, the citric acid cycle. So how do organisms fix CO2, reduce CO2? How do they make their own food? What is autotrophy? It is either the Calvin cycle, the reverse citric acid cycle, or the hydroxypropionate pathway. Just like with the Krebs cycle, I do not care about any of the intermediates. I don't care about the enzymes involved, right? We do want to keep in mind, like always, that whenever we see an arrow, there's an enzyme behind it, right? There's an enzyme there, which means there's a gene there, which means we can learn something about their genome. So all organisms who undergo this kind of autotrophy have something similar about them. They have the same genes. All the organisms that do this have the same genes. All the organisms that do this have the same genes. So if we're not focused on all of the little intermediates, what are we focused on, right? We're focused on the fact that in order to convert CO2, right, CO2 in, CO2 in, CO2 in, right, in order to do that, what do you need? You need two things. You need ATP, 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 lots and lots and lots of ATP, and you need electrons. So if you look at these pathways, right, so we'll take the most well-known first, right, Calvin cycle. To make a single sugar takes 18 ATP. That's a lot of ATP. To make a single 3-carbon molecule takes 5 ATP. To make this little molecule over here takes 3 ATP. It's energetically expensive to make food, right? Which makes sense because what are you going to do? You're going to take it somewhere and burn it apart. You can't get 38 ATP from a molecule of glucose if it costs nothing to make a molecule of glucose, right? No such thing as free money. So what we want to concentrate on is there are 3 ways that prokaryotes fix carbon. They are the Calvin cycle, the reverse citric acid cycle, and the hydroxypropionate pathway. I would just like you to recognize those terms. I would also like you to be able to acknowledge and to know that to make that happen requires energy and electrons. It costs a lot to fix CO2. That's what I want you to know. I don't care about how many ATP, how many electrons, how many sugars you make, that doesn't matter. These 3 pathways have in common that they all require electrons, they all require ATP in order to make food, right? So that is our big thing, right? They require ATP and they require electrons. That's what these 3 have in common. So I want you to recognize the name. I just want you to be able, if I show you a list, I want you to be like, that's autotrophy, that's autotrophy, that's autotrophy, that's not. And I want you to know it takes ATP and it takes electrons. Now, thankfully, we already know how they can get ATP, right? We spent a long time talking about that on Tuesday or Thursday. oxidative phosphorylation and chemiosmosis because we need a lot of ATP. Can we get a lot of ATP through substrate level phosphorylation? No. If we want a lot of ATP, what do we have to do, right? We have two options, two total, that's it. Substrate level, oxidative. Substrate gives me a little bit of ATP, oxidative gives me a lot. I already know I need a lot of ATP, so what do I need to do to make that ATP? Oxidative phosphorylation. I need to move electrons through an electron transport chain so that that will drive the pumping of protons so that those protons will move through an ATPase and make lots of ATP for me. So what we're going to be talking about is, where do you get the electrons and the ATP if you're an autotroph? Because what you do with them, they all had in common, right? What was the answer to question 3? I make food. What do I do with my electrons? I make food with it. Now we're going to go back to those questions, right? Where do you get your energy? Can't say they all have the same thing in common there. Two options, chemicals or light. Where do you get your electrons? You told me it's inorganic, but where? We're going to look at 3 places that you can steal electrons. One of them you already know because plants do it, and that's water. But we're going to look today at iron oxidizing and sulfur oxidizing because there are organisms who don't eat water, they eat iron. Right? You could say a plant, what does a plant eat? It eats water because that's its electrons. So you eat cheeseburgers, plants eat water. Some bacteria eat iron and some bacteria eat sulfur. Remember we talked about sulfur granules in chapter 4. And I said we're going to come back and we're going to talk in chapter 5 about why you would stockpile sulfur. And this is the answer, because that's their food. So for the same reason that someone would stockpile polysaccharides or fats, you would stockpile sulfur because that's your food source, right? So we know the answer to question 3, right? We're going to use those 2 things. We're going to use our energy in the form of ATP to pile the electrons onto CO2 and make food. We got that, right? All we have to remember is Calvin cycle, reverse citric acid cycle, hydroxypropionate pathway, boom, done. Now we're going to look specifically at our iron eaters, our sulfur eaters, and our water eaters and see how do they do this, right? How do you eat sulfur? How do you eat iron, right? So hopefully that sounds weird to you. How do you eat iron? Like, you know, grab rust and right? Like eat iron. So how do you eat iron? This is how you eat iron, right? So this is a lipid bilayer membrane. This is an ATPase. These are electron transport proteins. Does that sound familiar to you? Mm-hmm. All we have to do is get some electrons, and as long as we move them, then we'll translocate protons, and the whole system is set up, right? So here, they're eating iron. Fe2+. That's what they eat. Now, you can't follow the H's here, but you can look at the charge. Fe2+ becomes Fe3+. How did that happen? Well, they lose electrons, right? They lost electrons. So what do we say when something loses electrons? We say that it's been oxidized. So to eat iron means to oxidize iron, right? So you oxidize cheeseburgers, he oxidizes iron. He takes the electrons from iron, moves them through the electron transport chain, which translocates protons across and causes them to move through the ATPase. And you have your ATP, just like we had last week, but now we're not getting rid of our electrons. We're keeping them. So remember last week I was NADH, right? So NADH came over, and I handed Brent my electron. And it flowed through the electron transport chain, and Nassim was our oxygen. Today, what would actually happen in that electron transport chain is I, as NAD, would actually go down here, and I would sit at the end of the electron transport chain to collect the electron. Because we need it today. We didn't need it last week. Last week it was garbage, right? No offense, you're not garbage. But we got rid of Nassim, where I'm the mobile electron carrier. I'm the big guy, right, in the cell. So I'm going to go somewhere. Where am I going to go? The Kelvin cycle. So even though he's not a plant, that's how he fixes CO2, just like all the plants do. He makes ATP, He collects electrons, they go in and they get fixed. They are what fixes the CO2. So iron oxidizers do the latter part of this process just like plants do. They have the same Calvin cycle enzymes that plants have. The difference is where they get their electrons from iron. So we call this iron oxidation, which would have been foreign to you, but now that you know what oxidation works, you go, oh, oxidizing, that means we took electrons from them, right? That means that's their food. And these are really interesting guys. I mean, so here's an example. His name is Thiobacillus ferrooxidans, right? His species name is actually descriptive of the fact that he oxidizes iron, right? Because the Latin word for iron is ferrous. That's why iron is abbreviated. Its chemical symbol is Fe for ferrous. But look at how crazy this guy's life is. Notice, this is his plasma membrane. The only plasma membrane that a bacterium has is its cell membrane, right? So if we say outside, we mean the environment. Look at the pH of the outside of his environment. pH 2. That's like your stomach. And don't worry, he's not living in your stomach. There's no source of iron in your stomach. These guys actually live, the way where they were discovered was in these little streams. Like, this is actually a picture of rusty water. Rusty water coming out of an old mine, like a, what do you call that, an abandoned mine. Because there's a lot of ore in the rocks. And when they go and mine and they're looking for gold and things like that, they break rocks apart and the iron gets liberated and it goes into the water and it's acidic, and so it actually changes the water pH to like 2. And when they eat the iron, it turns it to rust. And so the water turns orange. So the water there is orange because there are so many iron oxidizing bacteria living there creating rust that it actually makes the water look like rust. Kind of fun, right? I think like miners in the 1800s in the United States, they would see that and they'd go, oh, that's yellow boy. There's yellow boy in the water. I mean, nobody knew what yellow boy was, but that was yellow boy. So who's yellow boy? Yellow boy is this bacterium who is oxidizing iron, turning it to rust, and using those electrons to fuel the fixing of CO2 to sugar, right? So that's where he gets his electrons. His electrons come from iron, so we say that he is an iron eater, right? There's where he gets his electrons, and he does not throw them away, he saves them. That's what everybody today is going to have in common. They don't throw their electrons away, they save them. All right, next up is our sulfur eater. The example of a sulfur eater that we have here is Sulfolobus acidocaldarius. So think about this. Acid, that seems pretty self-explanatory. Caldarius, think of like a witch's cauldron. So he lives in steaming acid. That's what that name means. These guys are typically found in deep sea hydrothermal vents, right? Where there's a break in the Earth's crust, and so hot material, right? Like molten rock is spewing out. It's got tons of sulfur in it, so they live in these very hot areas where sulfur is being excreted, right? And they like sulfur so much. Here's a picture, right? It's not the sulfur that's shiny here. That huge thing is a sulfur crystal, and they like it so much they actually grow right on top of it. And when they do that, what they're doing, right, is taking any number of electron-rich or reduced sulfur compounds, and they're oxidizing them. And when they oxidize them, right, so this is an example of this is elemental sulfur, which is what they're sitting on right there. Doesn't matter, we're just going to say sulfur, right? Incidentally, I don't care Fe2, Fe3, doesn't matter to me, right? Iron is a source of electrons. Sulfur is the source of electrons. Where do the electrons go? The same place they've been going since last Thursday. They go into the electron transport chain, through the electron transport chain, which generates the production of ATP because protons are being moved across the membrane. And then the big difference here, just like with the iron oxidizers, is we're keeping the electrons. We're not getting rid of the electrons, right? So where do we get the electrons? Sulfur. Where do they go? They go to fix CO2. Are you seeing a pattern? We're collecting electrons and we're using them to fix CO2. Now, in order to kind of try to make this meld a little bit with the heterotrophy, these you'll find with every single pathway that we've looked at. So if they're not helpful to you, they just make you more confused, just don't look at them. But if you want to be able to line up fermentation, respiration, iron oxidation, sulfur oxidation, and photosynthesis, then you can use these, right? So these guys are both chemosynthesizers, not photosynthesizers. Photosynthesizers use light energy to make sugar. These guys use chemical energy to make sugar. The chemo portion of that word means to convert chemical energy to ATP, right? So photo means to convert light energy to ATP, right? They use ATP and NADH, right? So this guy has iron as his source of electrons. Those electrons go into the electron transport chain, right? Cause the translocation of protons, which move through the ATPase and make ATP, and then they save those electrons, and the ATP and the electrons go into the Calvin cycle so that they can fix the carbon. And here's the thing that we don't want to forget. That's not for you. That's so that they can take the sugar over to another part of the cell and break it down for ATP. Which sounds ridiculous. You're like, seriously? Really? But even if it takes 16 ATP to make a sugar, how many ATP do you get for it when you burn it? 38. So you still get more than twice as much. As dumb as it sounds, because why don't you just keep the ATP? Why are you going through this whole process? You're making ATP, burning ATP, in order to make ATP again, right? But it's like an investment. You're investing a little bit, and you're getting interest on your investment, right? You're investing 16 ATP. It's like you putting money in the bank today and leaving it for the next 20 years, right? You leave it for 20 years, it's not going to be 16 bucks anymore. So if they make that, if they use 16 ATP today, and they invest it into this glucose molecule, by the time they get it over and break it apart in the Krebs cycle and completely oxidize it, now they've got 38 ATP. So they've made an investment in the molecule. Right? If you think about it, why does that sound so dumb, right? It sounds like you're doing twice the work. Well, you're getting way more than twice the return. You're using ATP today so that you have way more ATP ultimately. The other thing, right, those of you who have been in physiology know, this doesn't last for very long. You can't stockpile ATP. ATP will fall apart. So you need to invest it, right? It's kind of like my daughter finally figured out, like we were giving them an allowance in cash, right? And the cash just goes away. So she started giving me part of her cash allowance to put away from her, right? Like, just take this from me and don't give it back to me. Because she knows if it's in her wallet, she's at the grocery store, I'm gonna get some gum, let me get a frappuccino, and it's just, right, the money's gone. Do you ever do this? I hate having cash, because the minute I have cash in my wallet, it's gone. So you set it aside as something that will stay longer, right? You set that energy aside, not as ATP, which is gone, right? But as a sugar. And then you have the added benefit of when you take that sugar, which costs you 16 ATP, and you ultimately burn it, you get 838. So you get way more energy from it, right? So they do this so they can eat it. Their own heterotrophic processes, right? Because in every cell on the planet, you have glycolysis. Everybody does glycolysis. Not just you, but plants, and photosynthetic bacteria, and chemosynthetic bacteria. Everybody has that going on as well. So every organism that can do autotrophy is also doing heterotrophy. It's just they made their own food rather than eating it that someone else made. So that's iron oxidizing, right? So let's throw sulfur into there, right? So we have some kind of, and I put this one because it looks like water, it makes it a little easier to imagine, right? Some reduced sulfur compound, that's their food. What do you eat? I eat reduced sulfur. I steal the electrons from it. I send those electrons into the electron transport chain, generating ATP, saving the electrons as NADH, and then I take those electrons and I take that ATP and I make food, because that's my long-term storage, so I can burn it to make the ATP when I need the ATP. I'm not going to make 1 million pounds of ATP today, because by the time I need it, it'll all be gone. I'm going to take that ATP and fuel a long-term energy store process, right? So carbon fixation ends the autotrophy, but it doesn't mean that they just give it to you. That's not why plants photosynthesize. It's just that they make enough energy that they can tolerate us eating them and still survive because they've got extra. So that's chemosynthesis. Iron oxidizers, they eat iron. Sulfur oxidizers who eat sulfur. Now how about photosynthesis? Now for photosynthesis, we have to back up, right? We can't just say, where'd you get your electrons? Right? With an eater, with a heterotroph, where'd you get your electron, right? LB broth, TSB, whatever, motility media, right? The chicken broth that you're growing in, that's your food. Where'd you get your electrons if you're an iron oxidizer? I just ate iron. Where'd you get them if you're a sulfur oxidizer? I ate some sulfur. Where'd you get them if you're a water oxidizer? I ate water. So here's the thing about the electrons that fuel photosynthesis. They're weak little weenies that have zero energy inherent in them. Iron electrons, sulfur electrons, cheeseburger electrons, these are all already high energy that can go right into the electron transport chain. The electrons that are being used in photosynthesis are low energy, right? So if I'm the electron carrier who's going to be bringing it, it's almost like I just can't get there, right? I can't. I'm too tired. They're tired electrons. So we have to energize them. And we're energizing them using sunlight, solar energy. I was trying to do both of those at once, right? We're taking tired electrons and we're energizing them with the sun, which incidentally is what solar panels do, right? They take tired electrons and they energize them with the sun, right? And they learn to do that because they've looked at plants and they're like, huh, you can energize electrons with the sun? That's amazing, right? And then you don't have a $500 electric bill in the summertime. Not that I ever do, because I make my house stay at like 80 degrees. I'm like, I don't care if you're hot, go outside, take your clothes off, right? Because I pay the bill. When you pay the bill, you don't care anymore. You're like, put on a sweater when it's cold, I don't care. So, you use sunlight to energize, right? That's what photosynthesis is, right? That's the magic of photosynthesis. Somehow you took light and you converted it to chemical energy by energizing electrons. So it's still all about the electrons. It was all about the electrons here, we moved them through, we translocated the protons across. Now it's all about the electrons again, right? So here is my tired little electron. I stole it from water. It's tired, right? Oxygen wore it out because oxygen really likes electrons. So by the time you pull an electron away from oxygen, it is a tired electron. And you put it right here in the middle of this big radar dish. And you allow sunlight to shine on the radar disk, and that starts exciting electrons. And finally, right, it's like that thing at the fair. Did you ever go to the fair? And they got I should go sometime and ask them, right? But it's like you take a hammer and you whap it, and it makes this thing go up to the top, and it dings, right? And it's got all these like derogatory terms for what a wiener you are if you can't get it all the way to the top, right? The last time I went to the fair, I was dating someone who's not my husband because he's not like this. And the guy was like needling him because that's what they do. Like carnies like to tease you, right? So you give them $10 and play their stupid games. So he's like, I am such a wuss, right? And they did this huge thing, and I'm traumatized. Right? Don't go to the fair. But the idea there, right? If you're trying to energize this little piece of metal and shoot it up into the game and ding the bell, you're what's exciting that little piece of metal, right? You're using your mechanical strength to smack that down to resist gravity and send that thing up in the air. That's kind of like what the sunlight is here. The sunlight hits that electron and energizes it. And when it energizes it, it jumps up and it jumps up, and then it gets energized again because it's not quite there, and it jumps up again, and then it falls through an electron transport chain. So here I am, right? I'm not going to crawl on the ground because God knows what's on the ground, right? But I'm tired, and I get energized by the sun, and then I get energized again, and that allows me to come and bring the electron. So now I have an electron. Oh, I know what happens when I send an electron through an electron transport chain, right? Right now you've heard me say this 37,000 times, which by the way means that you should know it. You know when a teacher says something over and over and over and over and over again. Like when your mom says over and over and over again, pick your socks up. That means pay attention, right? So again, the electron goes into the electron transport chain. It moves through. The protons flow across. The protons go through an ATPase and generate ATP. That's the magic of photosynthesis. We have light energy at the start, and we have ATP at the end. Light, chemical. So what happens somehow, magically, right, the light hitting this magical, amazing pigment excites the electron. So we have a pigment here that is capable of absorbing light. That's what pigments do, right? And it's not just plants and bacteria. You know, you have pigments that absorb light too. They're all packed into your retina, right? So you're like, oh, plants, they're so magical. You can convert light energy into chemical energy. Guess what? That's what your eyes do. Light shines on your retina, and those little tiny light-absorbing pigments go, woo, that was exciting, and they send a chemical impulse to your brain, and you go, ooh, that was a flower, right? So you do the same thing, right? Any pigment, right, absorbs light. These guys, this chlorophyll, which we find in plants, absorbs light and excites electrons. And it excites them and allows them to go through an electron transport chain. Now, this kind of photosynthesis is what plants do. So it's probably what you saw in general biology. We call this non-cyclic either photophosphorylation or photosynthesis. So photophosphorylation is kind of a bigger word to wrap your mind around. So usually in general biology they say photosynthesis because it's easier. But photophosphorylation is actually more specific because what are you doing to ADP? You're phosphorylating it. How? Sunlight. So non-cyclic photophosphorylation, which is the technical term for photosynthesis, means a non-cycling electron fueled the addition of a phosphate group to ADP. And you're like, well, that's a mouthful, but that's what we just described. Here's an electron. It got excited, flowed through an electron transport chain, and that made ATP, right? Because there was an ADP and a P at the bottom end of that ATP synthase, and the protons flowing through, when it ratchet, boom. Phosphorylation, adding a phosphate group, right? Oxidative phosphorylation is the name that we give this. Now this is photophosphorylation, because it's fueled by light. So this right here, non-cyclic, because electron comes off of water, gets excited, flows through, and leaves. Not to be garbage, right? Where's the electron going in all of the things we're talking about today? To fix CO2, right? But it's leaving after one cycle. It does not cycle, so we call this non-cyclic photophosphorylation. You never said non-cyclic, maybe you did, or you might have called it Z-scheme photosynthesis, right? Because they call it sometimes that. Or maybe they just called it photosynthesis in general biology, because all you were doing was talking about plants. And that's fine. Plants, all eukaryotes, non-cyclic photophosphorylation. In fact, lots of bacteria undergo non-cyclic photophosphorylation. So if all you're doing is talking about plants, all you have to do is say photosynthesis. You call these the light-dependent portion of photosynthesis. Why? Because it depends on light. But we call this non-cyclic photosynthesis, right? Which probably, if there's non-cyclic, even if you couldn't see the other side of the slide, what would you think maybe there was? If there's non-cyclic, cyclic, right? So cyclic photosynthesis, so if I tell you non-cyclic photosynthesis, the electron goes through once and doesn't cycle, what do you think the electron does in cyclic photosynthesis? It cycles. This one is actually much more like that game at the fair where you whop it with the hammer, it goes up, and what does it do? It comes right back down to the same place. And you hit it again, and it goes up, and right back down to the same place. So this, electrons, right, get taken from this molecule, it becomes oxidized, goes here, light shines, excites it, and it goes through an electron transport chain and goes right back into that same pigment to get excited again. So what you'll see happen here is ATP, 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 lots of ATP being generated because as the electron's flowing through, what's getting translocated across? Protons. And then they're flowing through an ATPase, which is making ATP. And so every time that electron cycles, you make ATP. So you kind of stockpile a little ATP. Can you stockpile it forever? No, because it'll start to disintegrate. So ultimately, this will stop cycling and get donated to NAD. And now you have a bunch of electrons, you have a bunch of ATP, and you can fix carbon. So this is called cyclic photosynthesis or cyclic photophosphorylation. This is done in very few organisms. Most photosynthetic organisms do this right here. All the plants, all the algae, all the protists, all of the eukaryotes that photosynthesize do this. All of what are called the cyanobacteria do this. This cyclic photosynthesis that you see here, right, that's done in what are called purple sulfur bacteria. Can you imagine why they have the name purple sulfur bacteria? There's sulfur, right? What are they eating? Sulfur. Guess what color they are? Purple. Purple. They're actually kind of a reddish color, truth be told. They're red, right? Because why is a plant green? Chlorophyll, right? Why are cyanobacteria green? Chlorophyll. Why are these guys purple? Because bacterial chlorophyll is not green. Well, I should say it doesn't look green, right? Because of course everything looks like the colors it doesn't absorb, right? It's craziness that we don't need to talk about, right? But these guys are purple because they have a different pigment. So we call them purple sulfur bacteria. Now to make things crazy, we actually have green sulfur bacteria that are green, but they don't eat water, they eat sulfur. But if we were going to say, so we can say most carbon fixation is the Calvin cycle, with also hydroxypropionate and reverse citric acid cycle, and we could say most photophosphorylation is non-cyclic photophosphorylation. But we also have cyclic photophosphorylation. That's another way of converting light energy into ATP, right? You get your electrons, always non-cyclic photophosphorylation gets its electrons from water. Right, remember I said oxygen loves electrons, right? So you can imagine once we tear the electron off, it's super low energy and it will not ever get excited enough unless it's in this scheme where it can get excited twice. So non-cyclic photophosphorylation is always oxygenic photosynthesis. Why? Because it made oxygen. Cyclic photosynthesis is always anoxygenic. Because you're not using water, you don't make oxygen. You make sulfur, elemental sulfur. So what we can say, these live in areas where it doesn't smell yucky. These always live in areas where it smells yucky because they're always dependent on sulfur. So you're not going to find plants in Mono Lake or in the Salton Sea, but you will find these bacteria there because there's tons of sulfur there for them to eat, right? So let's apply that same crazy looking schematic, right? And if you don't like it, you don't have to look at them in your notes. But if you want to be able to compare fermentation, respiration, iron oxidizing, sulfur oxidizing, water oxidizing, or photophosphorylation, right? We had our electron donor, this craziness over here where we go through the electron transport chain, collect the electrons, produce ATP for carbon fixation. But notice this one, unlike every other one of these schematics that we've looked at, has this in the middle. This is absolutely important to all photosynthesis because that's the step where light becomes chemical. Why? Because you have a light harvesting pigment, they oftentimes call it, right? A light absorbing pigment that takes in light and excites electrons with it. So everything else was missing this step here because we just had an electron donor donating an electron directly to the electron transport chain, bringing it right over, right? But with photosynthesis, we have to energize the electron first, which means we have this, right? So the electron comes in, we put it onto chlorophyll, we excite the chlorophyll, and that allows it to donate it here, right? So photosynthesis, photo refers to light energy being converted to chemical energy, right, of ATP. And this produces for us the ATP and the electrons, either as NADPH, as in the case of plants, or NADH, as in the case of bacteria, right? It doesn't really matter whether you're NADPH or NADH, what do you have? Electrons. So it doesn't really matter, right? NADH, like that would be like if my sister came in here, she could be NADPH, right? Very similar, they carry electrons, it doesn't matter, right? We don't care whether it's NAD or NADP that comes, what we care is someone's coming and collecting the electrons so we can use it. So these guys, right, use the reducing power, the electrons and the ATP to fix CO2, right? Make it into a sugar. Here is a picture of some organisms that are microscopic that do the same kind of photosynthesis that plants do. These guys are all oxygenic phototropes. They use water, they oxidize it to oxygen, and they donate those electrons to chlorophyll, which gets excited and donates the electrons to the electron transport chain. Now, funny enough, they're not all green, just like all plants aren't green, right? And I'm not talking about plants in the fall, right, which doesn't happen in Southern California, but you know when you like look at pictures of the East Coast and like all the trees' leaves change to orange and then they fall out, and that doesn't happen here, right? But I'm not talking about that, like when the chlorophyll goes away. I'm talking about the fact that a lot of photosynthetic organisms who have chlorophyll just have other pigments, right? Some are green, like these cyanobacteria here, which are green like plants and green like other things. Some of them are kind of a brown color, like these guys here. These are called diatoms. This is a micrograph, not a scanning electron micrograph, just a light micrograph. So they're fairly large. They're actually really pretty. They have silica glass shells on the outsides of them. So they're really beautiful, right? You have these really beautiful geometric patterns. So this is a light micrograph. This is a light micrograph. That is a satellite image. I know we're terrible at geography, but maybe you recognize this boot-like peninsula at the bottom of Europe. It's called Italy, right? So this is a very big picture. This sort of milky area here, that is trillions and trillions of photosynthetic microorganisms called coccolithophores that bloom. Have you ever heard of an algal bloom? We had one this last spring, right, where the dinoflagellates bloomed and our water turned red, which is kind of gross. It's okay to swim in California. You don't want to swim in it anywhere else. But they're so abundant because fertilizer comes from the bottom of the ocean and light comes from the surface, from the sky into the surface of the water, and it's like you fertilize your garden and boom, they all grow, and they grow like crazy, and you can see them from space. Have you ever heard of a place called the White Cliffs of Dover? You could Google it, right? They're these huge cliffs, right? I don't know how tall, hundreds of feet tall, and they're white. Do you know why they're white? A bunch of these. Bazillions and bazillions of dead coccolithophores who have this calcium carbonate skeleton, and it's white. And when they die, they land on the bottom of the ocean, and they just pile up and pile up and pile up, and then an earthquake happens, and that piece of land comes up out of the water, and that's the White Cliffs of Dover. So you have some crazy photosynthesis going on in the oceans. In fact, we'll learn this later, in more detail. Half of all photosynthesis on the planet is microscopic. So we have rainforests, and they're wonderful, but half of all photosynthesis is done by organisms that are so small you couldn't see them with the naked eye. And most of them are doing it just like plants are, right? They're taking water, oxidizing water, and using the electrons from the water to pile them onto a CO2 and make food out of them. So that's oxygenic photosynthesis. Anoxygenic photosynthesis typically starts with a sulfur compound, oxidizes the sulfur, uses that, donates it to bacterial chlorophyll, which gets excited and donates it to the electron transport chain, right? And those bacteria, so here is a light micrograph. These are not stained bacteria. They're actually purple under the microscope. This is probably like 3000x. This is a very commonly studied organism whose name is Rhodospirillum rubrum. Rhodo means red. Spirillum means spiral shaped, right? So remember we talked about morphologies in chapter 4. These are spiral shaped bacteria. And if enough of them are growing, they can actually make the water turn purple. So these are purple sulfur bacteria. These are green sulfur bacteria, right? So you might at first glance go, God, they probably look very much like those do if they're really crowded. You know how you can tell the difference? Just take a big whiff. If you smell a bunch of sulfur, probably those do. The other thing that can help you is these guys are usually inhibited by oxygen, so they'll usually have to grow at the deeper portions of the water. Cyanobacteria don't mind oxygen at all, which makes sense because cyanobacteria make oxygen. It'd be super stupid if you made something that was then poisonous to you. But a lot of these guys, their pigment, the bacterial chlorophyll, is inhibited by oxygen. So if there's oxygen present, they can't photosynthesize, and if they can't photosynthesize, they can't make their own food, and if they can't make their own food, then they starve to death. So it's this really interesting thing where you'll end up with this sort of gradient on the surface of the water, right? Because remember, microorganisms are tiny, right? Remember we talked about motility? I said that microorganisms are small, and they have to experience life temporally. Right? They have to move somewhere and decide if now is better than it used to be. Whereas you and I can go, it looks like it's better over there if we go that way. Or I'm really hot and half of me is in the sun, so I'm going to move the whole of me into the shade. We experience spatially, they experience temporally because they're really tiny. So even in a chunk of mud that's on the surface of the water and is only like an inch deep, you actually have a stratification of organisms because here there's very little oxygen in that chunk. Here at the top, lots of oxygen. And so here at the top you see green stuff, right? And as the oxygen starts to decrease in concentration, you see those purple guys because they don't mind oxygen as much, and then as the oxygen continues to decrease, then you'll see those green sulfur bacteria. So you can see the vertical stratification, right? So they're moving within what you would say is the same environment. They're actually moving to teeny tiny little areas that are actually very different. The reason this green is not all over here is because this has too much oxygen and it kills it. So it's able to live just here. So this is a table that's been modified from the table in your textbook. So the one in your textbook is table 5.6. What I did was just removed a bunch of stuff that I don't care about. And I'm writing your test, so you shouldn't care about them. Not that you shouldn't care, but this is the stuff you should focus on, right? So, photosynthesis compared, right? So who is capable of photosynthesizing? Eukaryotes and prokaryotes. Eukaryotes would include members of kingdom Plantae, the plants, and members of kingdom Protista, the algae, right? Prokaryotes that can photosynthesize would include the cyanobacteria, the green bacteria, and the purple bacteria. They all have an electron donor. All of them have an electron donor that is inorganic. The difference, right? Water, sulfur compounds. Here's the nice thing. If you use water, what do you make? Oxygen. So knowing the answer to this one will tell you the answer to that one. If I use water, I make oxygen, so that's oxygenic. If I don't use water, I don't make oxygen, so that's anoxygenic. if I make oxygen, I must also live in an aerobic environment. Does that make sense? Because you're making oxygen, so you're creating the aerobic environment. So if you know the answer to this one, you know the answer not only to this, you know the answer to that as well. You use water, you create oxygen, you're an aerobe. You use something other than water, you don't create oxygen, and you don't live in an aerobic environment. What do you fix CO2 by? Safe bet is always Calvin-Benson cycle, because there are only very few organisms use that reverse citric acid, which is the reverse Krebs, or the hydroxypropionate pathway. Now these guys are kind of kooky. Cyanobacteria can actually do both of them. Cyanobacteria can use water, create oxygen, live in an aerobic environment, or if they don't have any water available to them to use, they can use sulfur. If they do that, they will not generate oxygen, they will not live in an aerobic environment, but they will still use the Calvin-Benson cycle. So they're versatile, which is kind of cool. Now, you probably feel like we've talked about, we've spent 2 days talking about metabolism, right? Which feels like you've done a lot. And you're like, she requires so much of us. So just in case you're feeling that way, rein it in, because this is, so you can't see it, right? Now I learned in school, I actually took classes on how to teach. I know not everybody does that, but I actually did. That's why I'm so good at what I do. And you're never supposed to say, I know you can't see this but I actually had a method to my madness. I don't want you to memorize any of this. So I know you can't see this, but remember what we said about arrows. Every time there's an arrow, there's an enzyme, and there's a substrate being converted to a product. This is the sum of all of the chemical reactions of a typical prokaryote, or photosynthetic prokaryote. That is what we have looked at. This is glycolysis, the Krebs cycle, and the electron transport chain. This is photosynthesis. See the little sum now? This is all the stuff I didn't make you learn. So see, it could have been a lot worse. So there's incredible diversity there, right? Lots of stuff, because that means that there's a ton of different things they can eat, a ton of different things they can make, right? Because every single one of those arrows usually is double-headed, which means they can take that pathway and go back and forth. Just like they can go Krebs or reverse Krebs. They can burn it to collect electrons, or they can build it, right? So incredible diversity. For instance, this handsome devil here, who is one of the non-sulfur purple bacteria, which is a total misnomer because he actually is a sulfur bacterium, he just doesn't use as much. So he can do all sorts of crazy stuff. If you put him in the presence of oxygen, you will keep him from photosynthesizing, which you would think would kill him, right? It's like if you took a plant and you put him in a box and kept it dark, what would happen to the plant? Sort of like Schrodinger's cat, right? Except for it's really dead. There's no philosophical ambiguity here, right? You can Google it. This guy will go, well, no, yeah, fine. Yeah, I can't photosynthesize. I will grow chemoheterotrophically. I'll be just like E. coli. I'll grow like E. coli. No problem, right? If you take away the oxygen and give him some sunlight, he'll go sweet, and he'll grow photoautotrophically. So just like we just learned here. So he will grow here, like we talked about on Thursday, or he will grow here, like we talked about today. So he can do both of those things. Can a plant do both of those things? No. Can you do both of those things? No. Can any eukaryote on the planet do both of those things? No. And you can give him not CO2, but other just kind of little garbage molecules, and he will actually make those into sugar too. So he's incredibly versatile, is all we need to say about that, right? I gave you details in case you were like, well, how versatile is he, Krista? That's how versatile he is. But what I want you to understand is, right, there's a way more going on here than we've talked about, right? Like I have a book, it's like this thick, and it has no pictures, and it is the biochemistry and physiology of prokaryotes. Super fun, right? You're not going to buy it. Hardly anybody buys it, right? But that's what, if you wanted to know the physiology and biochemistry of microorganisms, if you wanted to understand all of this, you would read a big giant textbook, right? Not just one chapter. So there's a lot going on inside individual organisms. And why do we care particularly so much in microbiology? Because we use all those little arrows to tell us who we've got in front of us, right? We can't see a lot about them, so we infer who they are by looking at what they eat. So the last half of this lab, starting with lab, I think it's 20, you will start inoculating your bacteria into all sorts of media, feeding them all sorts of crazy stuff. To see if you can eat it, you have the enzyme. If you have the enzyme, you have the gene, and now I know one thing about your genome. And I can look for all sorts of craziness, right? Just in this one single, like, seminal biochemical pathway of Krebs cycle, or glycolysis, Krebs cycle, and electron transport chain, right? That's just one of tons. But even in that one pathway, I can look to see what kind of proteins you can eat, what kind of amino acids you can fuel into there, right? So can you break apart proteins and bring them into glycolysis? Can you break apart amino acids and bring them into glycolysis or the Krebs cycle? What do you do with different polysaccharides, right? What do you do with different fats and other storage molecules? And do you have any other options besides oxygen as a terminal electron acceptor at the end of the electron transport chain? I can feed you all of these media and learn those things about you, and once I learn enough about you, I can determine who you are. And sort of the fringe benefit of that is we get to grow them in media that make them look really pretty, right? Like, this is called urea broth. Doesn't sound like it'd be pretty, does it? Urea, that's what you urinate, right? This is what color that broth turns when an organism can eat urea, and that's what color it is when it can't. So you can differentiate someone who can use urea versus someone who can't, or someone who can ferment glucose, or eat citrate, or eat fat, or use nitrate, or eat skim milk casein, right? All these different things you can learn by putting them into different media. So it's a particular point of interest for microbiology that we talk about metabolism, because metabolism is what we use for identification, right? We can't ask them who they are, we have to ask them what they eat, and that's how we can tell who they are. So that's why we pay so much attention to metabolism in general microbiology. And that is where we'll stop now. Let's take a 15-minute break, let the brains relax a little bit. And we'll start back up at 2:30. So if you wanted